so we're going to continue on with unit 2 rather, with section 4. Now this is a macromolecules unit, and it is a fairly lengthy section, or not unit, section here. So feel free to go back and watch this video, take your time, pause it, come back to it, whatever you need to do. But it is one of the longer sections that we will do, not only in this unit, but one of the longer ones in the class. All right, so the key elements to all living things. So living things are primarily composed of the following chemical elements: sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen, okay, or sponge, all right? So all living things primarily are composed of those elements. All right, so macromolecules. Think about what the word means, okay? So these are large molecules. Macro is large, micro is small. Macro elements or micromolecules are large molecules. They are in 4 classes in living things. So we're talking about carbohydrates, lipids, proteins, and nucleic acids. So macromolecules, all right, individual units of macromolecules are called monomers. Multiple monomers join together to form polymers. Polymers are formed by a chemical reaction called dehydration synthesis. So think about the term dehydration. When you think of dehydration, you think of lack of water or not enough water, okay? So dehydration synthesis is the joining of monomers by removing water. Okay? You're removing a water molecule to join monomers together. Polymers are broken into individual pieces by a chemical reaction called hydrolysis. Okay, so hydrolysis is the breaking apart of polymers into monomers by adding water. Okay, so dehydration synthesis, you are removing water to join monomers together. And in hydrolysis, you are adding water to a polymer to break them into individual monomers. So if you look at the pictures down here, you have an individual Lego. This would represent a monomer. Then you put a lot of Legos together to build a Lord of the Rings scene. You get polymers. All right, so the first type of macromolecule is carbohydrates. Right? More commonly, these are sugars. The monomer name of carbohydrates are monosaccharides. The polymer name of carbohydrates is polysaccharides. The primary function of carbohydrates is that it is the primary source of energy for all living things. Carbohydrates are composed of carbon, hydrogen, and oxygen in regular rations or ratios. So the amount of carbon equals the amount of oxygen, and the amount of hydrogen is always carbon times 2, or it's in a 1 to 2 to 1 ratio. For example, you have 6 carbons, which gives you 12 hydrogens, because it's carbon times 2, 6 times 6, or 6 times 2 is 12, and then the amount of oxygen equals the amount of carbon, so O6. C6H12O6 is sugar, okay? Now, this information here is going to be important. I'll go ahead and tell you for you to come back to as you do your macromolecules table assignment. All right, so carbohydrate storage. Let me make sure I did not skip a slide. I did not. Okay. Carbohydrate storage. So plants store long chains of glucose in polysaccharides that are called starch. Animals store long chains of glucose in polysaccharides called glycogen. And these are just 2 vocab terms. Structural carbohydrates. Cellulose is a structural carbohydrate that is used to build plant cell walls. Chitin is a structural carbohydrate that is used to build fungal cell walls and arthropod shells, like insects and crustaceans, or the lobsters that you see right here in the picture. All right, so lipids will be the next macromolecule that we go through. So examples include fats, oils, waxes, or steroids. Lipids are extremely nonpolar, which makes them hydrophobic. And remember, hydrophobic means water-fearing. Lipids are composed of long chains of hydrogen and carbon called hydrocarbons. These contain large amounts of energy. The monomer name is fatty acid. The polymer name is triglycerides. A triglycerides is three fatty acids connected by a molecule called glycerol. So tri meaning three, glycerides. Okay? The function is energy storage. Energy is stored in animals in tissues called adipose tissue, and also known as fat. So there's multiple different types of lipids. The first type is saturated fats. Saturated fats are completely full of hydrogen. What we mean by full of hydrogen is it's a single bond all the way around. Okay? And these type of lipids are solid at room temperature. Then you have unsaturated fats, and these are able to receive more hydrogen because of the carbon-carbon double bonds. Monounsaturated fats are fats that contain one carbon-carbon double bond, and polyunsaturated fats are fats that contain multiple carbon-carbon double bonds. So right here you see a carbon-carbon double bond, and this would be a monounsaturated fat. Okay. Hydrogenated fats are unsaturated fats that have had hydrogen artificially added to them, thus making them solid at room temperature. The filling of a Twinkie, the cream filling of a Twinkie, is a great example of a hydrogenated fat. So phospholipids are lipids that contain a phosphate group and they compose the cell membrane. So next unit, we're going to do a lot of work with phospholipid bilayers, the cell membrane. Part of them are hydrophobic, part of them are hydrophilic. Hydrophilic's water-loving, hydrophobic's water-fearing. Some of those words you already know, some you don't. Okay, but lipids compose the cell membrane. Then you have waxes. So a wax is a lipid plus an alcohol, and lipstick is an example of a wax. Now steroids serve as a communication molecule that can be distributed throughout the body. Cholesterol is a special steroid that gives the cell flexibility. Then we'll move into the third type of macromolecule, which is proteins. The monomer name of proteins is amino acids. The polymer name of proteins is proteins or polypeptides. These two names are used interchangeably. Okay, so it can be proteins, it could be polypeptides, it could be both in the same sentence. Proteins are essential in almost every cellular function. I'm going to go ahead and tell you now. You're going to hear proteins come up for the rest of the year. Okay, proteins are extremely important, and we're going to revisit them a lot. So go ahead and learn proteins now. Some things that proteins are used for is structural, signaling, catalyst, immunity, regulation, poisons, and transport. Okay, so they build things, muscles, skin, and hair. Insulin is a protein. Enzymes, which we'll talk about in the next section of this unit. Antibodies, body processes, venom. Okay, snake venom is hinged on or dependent on proteins. And then when we get to cellular functions and the enzyme substrate complex, we'll talk about cell transport. And you may need to pause the video here to if you're working on your macromolecules chart right now. All right, so proteins have 4 levels of structures. You have primary structure, secondary structure, tertiary structure, and quaternary structure. This next bullet is all caps, and it's because it's important. It's going to be something that comes up throughout this entire class. Protein structure determines what its function is. So how a protein is put together will tell that protein what job it's supposed to do. And this is where we see a lot of health issues come into play by a protein that is not shaped right. Okay. So when we get to genetics, which will be a little while, but it will play into to protein structure once we get to genetics. So the primary structure of proteins is just a string of amino acids. Okay, so if we look at A, this is the primary structure. The secondary structure is a helix structure formed from primary structures. So you have some primary structures put together in a helix or a pleated sheet type structure. The tertiary structure is the secondary structures that is folded on itself. This is a completed protein. So you take the helix and you fold it up on itself, and this will be the tertiary structure. And this will be, like it says, a completed protein. And the last one, quaternary structure, is multiple tertiary structures together. Not all proteins have this level of structure. So you see down here, you have, in this particular example, we have 4 different tertiary structures together for this quaternary structure. All right, so protein denaturation. Go ahead and learn the vocab term denaturation right now. It is important. So this is when a protein loses its structure due to being exposed to something. That something is heat or high or low pH. Okay, both of these can cause a protein to denature. This will cause a protein not to do its job. When we get to talking about homeostasis, we'll talk about fever, okay? Temperature, increase in temperature. When you're sick, you have a fever. There's a reason. There's a reason that's part of your body's immune system response, and it deals with protein denaturation. And the last macromolecule is going to be nucleic acids. Nucleic acids is DNA and RNA. The monomer name of nucleic acids is nucleotide, and there's not a polymer name. Functions of nucleic acids is to store hereditary information. DNA codes for proteins. DNA contains the information to make every protein in your body, which in turn is going to make your body function the way that it's supposed to. Nucleotides are made up of 3 components: a phosphate group, a nitrogenous base, and a 5-carbon sugar. The 5-carbon sugar, is the primary structure, or primary difference between RNA and DNA. In DNA, it contains the sugar deoxyribose. In RNA, it contains the sugar ribose, hence where the D and the R comes from in those abbreviations. And we'll get into more of that as we move through the class as well. So that's where I'm going to end this video. Again, feel free to go back, rewatch it, pause it, use it to help you study and fill out your assignment. If you have questions, let me know.